In 1902, Gottlieb Herberland, an Austrian botanist, developed the concept of in vitro cell culture, or we also call it plant tissue culture. He cultivated isolated plant cells in vitro of an artificial grow medium. The plant cell in his experiment increased in size but failed to divide, due to which his prediction failed. However, his hypothesis paved the way for more research. He is thus regarded as the father of tissue culture. Taking cue from Gottlieb's experiment, Philip R. Wright experimented on tomato roots. He reported not only the culture of tomato root tips could grow, but they could also repeatedly subculture to liquid medium of inorganic salt supplemented with yeast extract. In 1963, White had developed the White's media that's specifically used in root culture. A breakthrough came from Falk Carl Cook and Toshio Murashige. These two had developed a growth media named Murashige and Cook media. At the beginning, this media was applied for in vitro callus culture of Nicotiana tabacum, but it is found appropriate for most of the plant species. This MS media contains a high concentration of nitrate, potassium, and also ammonia. The plant part used in culture is called explant. The explant can be apical meristem, leaf, petiole, axillary bud, node, and also internode. But don't forget the plant root system that also can be an explant too, such as root apical meristem, root tip, or root hair. Is then placed in a suitable nutrient medium containing sucrose, inorganic salts, vitamins, amino acids, and plant growth regulators. The explants are allowed to multiply and form a group of tissues called callus. Callus is an irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus will be subcultured into appropriate media to induce the growth of embryos. The embryos is development of embryo derived from somatic cells under an appropriate artificial environment. This embryo then will be transferred or subcultured into growth media for induction of root and shoot to form many plantlets. These plantlets are then transferred into different containers for rapid multiplication. Finally, the developed plantlets are going through hardening and acclimatization process before transferred into the greenhouse or to the soil for the normal growth. In the plant tissue culture laboratory, you can control some parameters to get a healthy plantlet, such as relative humidity, light intensity, the suitable temperature based on the type of plant, and also the time duration of plant incubation. There are many advantages of tissue culture technique, which are as follows. First, it can give you high yield crop. The second one, it can produce a disease-resistant plant and also pathogen-free plant or crops. Besides that, rapid growth of multiplication uniform plants can be achieved through macropropagation method. Besides that, these techniques are able to supply the consumer market with new variety. Thank you for watching, and I hope you all enjoy my video. Goodbye.